my topic for this afternoon will be salivary glands and pancreas. Now, these salivary glands and pancreas are both glandular epithelium and part of the digestive system of the body. But before we go into the glands, let us first have a review of the glandular epithelium. So under the glandular epithelium, let's review about glandular histogenesis, both the exocrine and endocrine glands. So let us start first with glandular histogenesis, or how are glands formed in the body. So every cell in the body will have to undergo the 4 stages of cytomorphosis. These 4 stages are the following. We have the embryonal stage. Second, we have the stage of specialization and maturation. The third is the stage of regression and senescence. And the fourth stage is cellular death, or the death of the whole tissue. So, during the second stage, the stage of maturation and differentiation, if group of cells will form a particular structure or organ, since we have the glands, the cells are mostly epithelial cells. So this group of epithelial cells will undergo proliferation and they will grow inward, or there is what we call the process of involution. Since we have 2 types of glands in the body, there will be 2 processes that will be involved. For exocrine gland, the cells that have undergone proliferation will start to form a gland inward. So there is an attachment from the surface cells to the gland, and this attachment will now be formed into excretory duct, and some of the cells will form now the secretory cells of the gland. For the endocrine gland, there will be detachment from the surface cells, so the group of cells that are formed will settle deeper into the tissue and will be located in the blood vessels of the tissue, forming into an endocrine gland. So in the 2 types of glands, so for the exocrine glands with the excretory duct, the substances that are formed by the gland cells will pass through the passageway or through the excretory duct. While in the endocrine gland, the substances that are formed by the gland or by the secretory cells will pass directly into the blood circulation. That's why they are found near blood vessels in the form of cords, of islets, and form of clumps of cells. So for the 2 types of glands that are formed, we have the exocrine glands and endocrine glands. So for the 2 types of glands that are formed, we have the exocrine glands and endocrine glands. So take note, exocrine glands with ducts or passageways, while the endocrine glands in the form of cords, in the form of islets of cells or clump of cells near blood vessels, and is considered to be ductless. So for the exocrine glands, the glands produces and secrete substances onto an epithelial surface by way of a duct, either to the surface of the body. So this is exampled by sweat glands and sebaceous glands or to the lumen of the digestive tract. We have the gut and the digestive glands. The exocrine glands are composed of a cluster of secretory cells which collectively form an axinus or axini. The axini are surrounded by a basement membrane and are held together by tight junctions between secretory cells. And these cells possess a highly developed endoplasmic reticulum and Golgi network for material secretion and are rich in mitochondria. So it's not only endoplasmic reticulum and Golgi bodies, but also mitochondria. Let's have a closer look of the exocrine glands. So let's start first with the secretory portion of the gland. So the secretory portion are composed of secretory cells constituting the parenchyma of the gland. And these secretory cells are either grouped as tubules with tubular structure or maybe sac-like structure which we call axini or alveoli. The glandular cells vary according to the kind of secretion and stage activity of the cell. So in the illustration, we have a sac-like formation of the secretory portion, and we have the secretory cells, and these cells are responsible for synthesizing substances and releasing these substances through a duct system, being an exocrine gland. The excretory portion of the exocrine gland is the excretory duct. The excretory duct may be branched or may be unbranched. This duct conducts glandular products to a free external or internal body surface. Now, in some larger or major exocrine gland, the main ducts may divide into interlobular ducts located in the connective tissue trabeculae, separating into lobes. From the interlobar ducts, it may branch into interlobular ducts located in the trabeculae between the lobules. From the interlobular ducts, these are continuous with the intercalary ducts whose branches communicate with the secretory axini. The epithelium of the largest duct may be simple or stratified columnar cells. As ducts become smaller, there is a change from simple cuboidal cells at the terminal portion, finally becoming simple squamous cells. Both the secretory and excretory portions of exocrine glands are supported or invested in loose areolar connective tissue stroma containing numerous blood capillaries. The secretions of the exocrine glands are the following: pure protein products or enzymatic proteins. The secretory cells releasing this type of substances are called serous cells. So these are best exampled by the axonar cells of the pancreas and parotid glands. Then the exocrine glands also releases mucus, or they can secrete also mucous substances. And these are best exampled by goblet cells, mucous neck cells of the gastric glands, and glands of the respiratory gland. Then we also have substances secreted by special exocrine cells, and the product is released through a particular process of extensions, such as the sebaceous gland secreting sebum, the mammary gland secreting milk, and the parietal cells of the gastric glands secreting hydrochloric acid as chloride and hydrogen ions. The next two slides will give us the illustration of the exocrine gland. So in this illustration, we have the duct of the exocrine gland and we have the secretory cells. So take note of the cells composing the duct of exocrine gland and the shape of the cells composing the exocrine gland. So we have the passageway of the exocrine gland and we have the secretory cells containing mitochondria, vesicle, and endoplasmic reticulum. Again, we have the illustration of the exocrine gland together with the cells composing the duct and the glandular epithelium. So, the secretory cells or the secretory units of the exocrine gland produces substances that are elaborated to the surface via the ducts, and the shape of the cells in the secretory portion are more or less columnar to pyramidal cells. Then into the striated duct, it could be cuboidal cells or columnar cells going to the outer part of the cells. So the duct is part of the surface cells, giving the passageway of the substances produced by the secretory cells of the exocrine gland. From exocrine, let's go into the endocrine glands. So the endocrine glands, these are epithelial cells arranged either in cords, bundles, or islets, or just a group of cells invested in connective tissue stroma containing numerous fenestrated blood capillaries. The secretion of endocrine glands includes polypeptide or pure protein hormones, glycoprotein hormones, steroid hormones, and biogenic amines. So we have here illustration of exocrine glands and endocrine glands. So try to focus on the illustration and take note of the differentiating points between the two glands. Let's go into the classification of glands. So the first criterion is based on the number or types of component cells. So under this criterion, we have unicellular gland. This is a one-cell type gland represented by goblet cell. From unicellular, we have the multicellular glands composed of two or more types of cells. So the simplest form is a sheet of epithelium consisting of a homogeneous population of secretory cells as represented by the illustration of the gastric mucosa. The next criterion is based on the morphology of gland or the shape of the glands. So we have the tubular gland and we have the sac-like structure, we have the alveolar glands. So again, based on the branching of the duct, we have the simple glands and we have the compound glands. So we have the simple tubular glands, simple branch tubular glands, and we have the compound tubular glands. So we have an example also of the location of the glands in the body. Then we have the alveolar glands, we have the simple alveolar glands, and we have the simple branch alveolar glands, and we have the compound alveolar glands, and a combination of the compound tubulo-alveolar glands. So a mixture of the tubular glands and alveolar glands. The next criterion is based on histological characteristics of the secreting cells. So we have the serous glands wherein the secreting cells are acidophilic. So the cytoplasm contains eosinophilic granules with round nucleus and wide lumen. So this is best exampled by the parotid gland and the pancreas. So in the illustration, you can see the serous gland with the acidophilic cytoplasm, round nucleus, and the lumen. The mucous glands, the mucous glands are basophilic in nature or in staining property. So the secretory cells of the mucous glands, they are basophilic, non-granular, reticulated cytoplasm, and a flattened nucleus towards the basal surface with wider lumen. So this is best exampled by the sublingual glands. The Mucoserous glands or mixed glands have secretory cells which are both serous and mucous type of cells, so the secretions are both watery and viscous. And this is best exampled by the maxillary glands. The fourth criterion is according to the nature of secretion. So we have again serous gland, mucous gland, mucoserous gland, and cytogenic gland. So serous, watery secretion, mucous, mucinous secretion or viscous secretion, mucoserous, a mixture of mucus and watery secretion, and we have the cytogenic gland, a gland that produces cells. So we have the testes, we have the sperm cells, and the ovary, we have the egg cell or the oocyte. Let us take a closer look at the mucous cell. So the mucous cell secrete a thick viscous substance or viscous mucoprotein which we call mucigen. Mucigen plus water, we have mucus. The function of the mucus are the following. It coats food, making it easier to transport food from the esophagus to stomach. It protects food from the digestive enzymes released by the gastrointestinal tract. It traps foreign materials in the respiratory tract. So the shape of the mucous cell, it is roughly pyramidal in shape with flattened nucleus next to the basement membrane or towards the basement membrane. So we have here in the illustration, on the upper portion we have a mucous cell, and in the lower illustration we have mucous glands. So these are the minor salivary glands of the palate. Okay, so we have the secretory cells of the mucous gland. The serous gland is composed of serous cells. So a serous cell is pyramidal in shape and it secretes watery product containing enzymes. The nucleus may be spherical to sub-spherical in shape. And in the apical part of the cell, we have secretory granules that are involved in the secretion of the serous cell. We have the mixed gland. It is also referred to as serous demi-lune. It both secretes mucus and watery products into the saliva. The parenchyma cells of these glands may be serous or mucous cells or both. In this case, a cluster of serous cells form a half-moon-shaped structure on the surface of mucous cells referred to as serous demi-lune. To differentiate the three glands, mucous glands, serous glands, mixed glands, so we have here an illustration. Take note of the shape of the nucleus, the location of the nucleus, the staining property of the cells involved, and the lumen of the gland. The fifth classification is according to the integrity of the secretory cells or manner of elaboration of secretion. So we have the merocrine gland, the apocrine gland, and holocrine gland. In the merocrine gland, the secretory cells are intact. So the secretory cells are not involved or not part of the secretion. While the apocrine gland, the secretory cells are partially involved in the secretion. While in the holocrine gland, the whole secretory cells are involved in the secretion. In the sixth classification, it is according to the presence or absence of excretory ducts. So we have the endocrine glands and the excretory glands. So endocrine glands, they are ductless glands and they are found in the pituitary, thyroid, parathyroid, adrenal or suprarenal glands, pineal glands, and some neurons in the hypothalamus of the diencephalon of the brain. The excretory glands have excretory duct system. Let us now go into the salivary glands of the body. So we have already a background of the different glands in the body, and in the salivary glands of the body, let's try to see what type of glands are involved. Salivary glands are structures that produce products that facilitate transport and digestion of food within the gastrointestinal tract. We have here the structural and functional unit of salivary gland. So the secretory part of the gland is what we call axinous, and take note the functional and structural unit of salivary glands. So it is composed of secretory cells with myoepithelial cells involved in the secretion and a basal lamina. From the axinous or the last part of axinous, we have a change of the cell from pyramidal shape to cuboidal shape, giving the passageway of the salivary gland, which is referred to as intercalated duct. And this is also associated with myoepithelial cells going to the upper duct. So from the intercalated duct, there is a change again of cells from cuboidal to columnar. We have the striated duct of the salivary gland. Take note, near the secretory cells are the intercalated duct followed by striated duct. The salivary glands are categorized into major salivary glands and minor salivary glands. For the minor salivary glands, these are located in the mucosa of oral cavity. They open directly or via short duct onto the surface of the oral epithelium. The minor salivary glands secrete continuously, contributing saliva that moistens and lubricate the oral cavity. So, the Ducts are relatively short, branching tubules lined with mucous cells. The minor salivary glands are around 6 million bilaterally and are located all over the oral cavity except the following: gingiva, anterior raphe of the hard palates, anterior two-thirds of the tongue. The minor salivary glands are the following: labial and buccal glands. We have palatine glands. In the tonsillar areas, we have the Weber's gland. In the molar, retromolar area, we have the Carr-Maltz glands. And anterior lingual salivary glands, we have the Blandine and Noon glands. And postlingual salivary glands, we have the von Ebner's gland. There are three major salivary glands. We have the parotid glands, the submandibular glands, and we have the sublingual glands with branching system of ducts. So you can see the location of the major salivary glands in the illustration. Another illustration of the location of the different major glands of the body: parotid gland, submandibular gland, and sublingual gland. The parotid glands is the largest major salivary glands. It is located between the ear and the jaw and is a serous gland. Secretion is purely serous in nature. The duct is referred to as Stenson's duct. The submandibular or submaxillary salivary gland is located under the jaw, and it is classified as a mixed gland. The predominant secretion is serous, and the duct is referred to as Wharton's duct. The sublingual salivary glands is located under the tongue or located at the floor of the mouth. It is also classified as a mixed gland. The secretion is predominantly mucous, and the duct is referred to as Bartholin's duct. For the secretions, we have the pure serous secretions secreted by the PP glands, parotid glands, and posterior lingual glands. And for the pure mucous secretion, we have the GPA, palatine glands, and anterior lingual glands. For the mixed glands or mucoserous glands, we have the sublingual, which predominantly secretes mucus, submandibular, which predominantly secretes serous secretions, and labial and buccal glands, predominantly mucous secretion. You might ask, what is that red L in sublingual? That is just to remember as the type of secretion. So what comes after L is M, so predominantly mucus. And in submandibular, we have the R. What comes after R is S, so predominantly serous secretion. Okay, so that is the purpose of the L and the R. So we have here an illustration of the system of ducts. So just get familiar of the formation and pathways of this duct system so that we can trace the secretion of the substance produced by the gland. Another illustration of the system of ducts, this time assisted by arrows. So on the lower left side, we have the different duct system from striated duct to intercalated duct, then serous cell or mucous cell. So take note, after the parenchyma or secretory cells, we have the intercalated duct. Then going upward, we have the striated duct. We have here different types of axini in salivary glands. So we have the serous, the mucous glands, and the mucoserous gland. This mucoserous gland is also referred to as serous demilune or crescent of Giannuzzi. Giannuzzi is the histologist credited for this gland. So you can see in this illustration, we have the S, which means serous, and we have the M, which means mucus. So in this type of gland, you can see the difference between a serous gland and a mucous gland, which is also illustrated above. So take note of the location, the shape of the nucleus in the serous gland compared to the mucous gland, which is flattened and found on the basal part of the cell. These major glands produce a large volume of secretion on mechanical, chemical, or olfactory stimuli. The saliva in these glands is a mixture of the products of the minor and major glands. The saliva is a viscous, colorless, opalescent fluid containing the following: water, mucoproteins in the form of enzyme thiolane, we have immunoglobulins, we have inorganic ions such as calcium, potassium, sodium, chloride, and traces of iron, and some salivary corpuscles. The salivary corpuscles are degenerating granulocytes and lymphocytes from tonsils and lymph nodes at the back of the tongue. We have here the microscopic appearance of a parotid gland, and take note, the parotid gland is a serous type of gland. So take note of the color of the cells, which is pinkish in color. So the cytoplasm is more or less pinkish in color, being acidophilic, with a lumen and with the nucleus, which could be round or which could be oblong in shape and could be found either at the center of the cell or towards the basal part of the cell. We have here another illustration of the parotid gland, which is acidophilic, and it has a secretion which is described to be watery serous saliva rich in amylase or proline-rich protein with a duct system referred to as Stenson duct. The submandibular gland is more or less a mixed gland, but the secretion is more mucinous than watery, and the duct system is referred to as Wharton's ducts. So take note again of the appearance of the gland under the microscope. Then we have here the sublingual gland, which is also a mixed gland. So mucous acini gland or cells with serous gland. Now, the sublingual gland, the secretion of the sublingual gland is 60% mucous and 30% serous. So we have here again the different microscopic appearances of the different major glands of the body. So the parotid gland contain only serous acini. The submandibular gland and sublingual gland, being mixed gland, contains both the mucous and the serous acini. The submandibular gland has more serous acini compared to the mucous acini, while the sublingual gland are composed mostly of mucous acini and some serous acini. We will now go into the organs involved in digestion. So the organ assigned to me are the pancreas, and we will describe the pancreas both anatomically and histologically. The pancreas is a pinkish white organ lying retroperitoneally on the posterior wall of the abdominal cavity at the level of the second and third lumbar vertebrae, or simply the pancreas is found behind the stomach. It is about 10 inches in length or 20 to 25 centimeters in length, with 2 inches width and almost 100 to 150 grams in weight. The average shape of the pancreas, it looks like banana. So it has a banana shape appearance, or it is referred to as a banana shape organ of digestion. So we have here the illustration of the pancreas. It is composed of the following: the head portion, the neck, the body, and tail. So narrower going upward. So the head is lodged in the concavity of the C-shaped duodenum. Narrower body and tail extend transversely across the posterior wall of the abdomen to the hilus of the spleen. The pancreas is protected by a thin layer of connective tissue. It is lobulated and described to be the second largest gland associated with the alimentary tract. So it is a compound actinous gland made up of many lobules bound together by loose areolar connective tissue. The pancreas has both exocrine and endocrine portion. The exocrine portion secretes enzyme-rich fluid required for the digestion of fats, carbohydrates, and proteins. For carbohydrates, we have the pancreatic amylase. The endocrine portion secretes hormones essential for the control of carbohydrate metabolism. We have here the appearance of the pancreas. On the upper portion, we have the anatomical appearance of the pancreas, and in the lower portion, we have the microscopic appearance of the pancreas taken in the narrower portion of the pancreas towards the tail. So take note, in the microscopic appearance, there are two types of glands that can be seen. We have the serous gland and we have the mucous glands. So the serous glands, we have the pinkish cytoplasm, and the mucous gland, we have the blue cytoplasm. Again, describing the pancreas is a mixed gland consisting of an exocrine and endocrine portion. Exocrine portion secretes pancreatic juice and endocrine portion secretes hormones. Again, we have illustration of the pancreas, anatomical appearance and microscopic appearance. So upper portion anatomical appearance and lower portion we have the microscopic appearance, both the serous and the mucous glands. Illustrated again. The pancreatic axini is serous in nature, lined by pyramidal cells and have small lumen. The nucleus in the axinar cells, we have the supranuclear region filled with zymogen granules, and the infranuclear regions, it is intensely basophilic. And the duct system are the following: intralobular ducts, interlobular ducts, and the main duct. The axinar lumen may show pale staining cells of intercalated duct referred to as centroaxinar cells. So in the illustration, you can see the intercalated duct and the secretory cells. The secretory portion of the endocrine pancreas is in the form of islands. So we have the islets of Langerhans, which is lightly stained using the hematoxylin and eosin stain, and they are scattered among the axini of the exocrine part. So we can differentiate under the microscope just by the color of the cells. The cells of the islets are arranged as anastomosing plates, and the islets contain three types of cells: alpha, beta, delta cells, which are seen with special stains. We have the illustration of the islet of Langerhans. On the surface part of the islet, we have the exocrine pancreas composed of axonar cells and duct cells. Towards the islet, we have the three types of cells: alpha cells towards the exocrine pancreas, which secretes glucagon, and towards the middle, we have the beta cell, which secretes insulin, and scattered along the alpha and beta cells, we have the delta cells which secretes somatostatin. So you can see the different colors of the different cells composing the islet of Langerhans. The islets of Langerhans are scattered throughout the pancreas, and it is more numerous in the tail portion of the organ. Just an estimation, over 1 million islets in the human pancreas, but owing to their small size, constitute only 1 to 2% of the volume of the gland, and 2 to 3,000 cells per islet arranged in anastomosing cords or plates. Histologically, the islets is described as a compact mass of epithelial cells pervaded by a labyrinth network of capillaries, and it is demarcated from the surrounding axonar tissue by a thin layer of reticular fibers that extend inward to form a delicate investment around the capillaries. The hormones that the islets secretes are demonstrated in the laboratory using fluorescein conjugated antibodies for definitive identification of their cells of origins. And all of the hormones secreted are involved in the control of the level of glucose in the blood. The beta cells are the predominant cell type in the islets, mostly found at the center of the islets, making up 70% of its mass. The beta cell secretes insulin, a polypeptide with 21 alpha chain amino acid and 30 beta chain amino acid linked by 2 disulfide bridges. The insulin is an essential hormone directly or indirectly affecting cell function in nearly every organ of the body. It stimulates all body cells to take up glucose except the brain cells. So, it is considered to be a cell wherein there is glucose built up and as a storage for glucose in the body. So we have here an illustration of the beta cells, which is more towards the tail portion of the pancreas. So in the islets of Langerhans, it is mostly found towards the center of the islets and is responsible for the release of insulin in the body to control the blood sugar. The alpha cells secretes glucagon, and they are located mainly at the periphery of the islets, and the glucagon is considered to be the antagonistic hormones, and it is secreted by the alpha cells in response to a fall in blood glucose concentration, which has an effect on the liver cells. It causes the breakdown of glycogen to release glucose into the blood. So when glycogen is depleted, glucagon can enhance glyconeogenesis by the hepatic cells or in the hepatic cells. The delta cell secretes somatostatin, which is a small polypeptide of 14 amino acids. Secretion is stimulated by postprandial increase in blood glucose, increase in amino acids, or increase in fatty acids. Its local effect in the islets are inhibitory, decreasing the rate of secretion of both insulin and glucagon. Present also in the islets are the F cells, but they are present in very small numbers, and it secretes pancreatic polypeptide. That's why they are called PP cells, pancreatic polypeptide. Little is known about its function or the control of its release. The exocrine pancreas is covered with a capsule, wherein from this capsule arise septas, which divide the gland into small lobules. So the interlobular is made up of connective tissue, which contains large ducts, blood vessels, and nerve fibers. And the connective tissue also surrounds the axini, small ducts, and islets of Langerhans. The exocrine pancreas is a compound axinous gland. The axini are round or slightly elongated, consisting of 40 to 50 pyramidal epithelial cells around a narrow lumen. The size of the lumen varies with the physiological state of the gland, becoming wider during active secretion. The cytoplasm of the exocrine pancreas are strongly basophilic near the base of the axonar cells because of the presence of the increased amount of ribonucleic acid or RNA. In the apical part of the cell, the cytoplasm is filled with large numbers of secretory granules or vesicle granules or zymogen granules. We have the duct system. Okay, so take note of the different ducts composing the gland. So from the axini, we have the centroaxonar cells, which appears to be low columnar to squamous cells, forming now the intercalated ducts. Then from intercalated ducts to intralobular ducts to interlobular ducts, then into the two main pancreatic ducts, the duct of Versong and the duct of Santorini. Now, the duct of Versong is found in larger glands, which begins in the tail, running through the length of the gland, gradually increasing in diameter as it is joined by the numerous interlobular ducts. We have the duct of Santorini, but this duct is an accessory duct. So the main duct system of the gland is the duct of Versong. And this is the last slide. Thank you for your patience.